Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about clinical management in Friedreich ataxia. Many physicians and individuals with FA consider vitamin and supplement regimens. Clinical studies have suggested modest or subjective benefit, but none have been proven. Among those taken are vitamin E, coenzyme Q10, and idebenone. Committed international physicians and researchers have critically evaluated medical literature, outcomes research, and clinical practice for treating symptoms of FA, and have come together to prepare the first comprehensive consensus clinical management guidelines. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about how genetic mutations cause Friedreich's Ataxia. FA is an inherited or single gene disorder. Mutations or DNA changes in the FXN gene cause FA. FA is inherited in an autosomal recessive manner, meaning that individuals with FA have two mutated or abnormal copies of the FXN gene. This means both biological parents must be a carrier of the disease for a child to be affected. It is estimated that 1 in 100 people are carriers, and carriers do not exhibit symptoms of FA. Each such carrier parent has one mutated gene, allele, and one normal gene, allele, in the FXN gene. Because each child gets one of the mother's genes and one of the father's genes in this location, there are 4 possible combinations of the genes passed down to the child, or a 25% chance that the child will have FA. The FA gene mutation limits the production of a protein called frataxin. Frataxin is known to be an important protein that functions in the mitochondria, the energy-producing factories, of the cell. Frataxin helps to move iron and is involved with the formation of iron-sulfur clusters, which are necessary components in the function of the mitochondria and thus energy production. We also know that specific nerve cells, neurons, degenerate in people with FA, and this is directly manifested in the symptoms of the disease. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about genetic testing in Friedreich's Ataxia. Genetic testing analyzes the FXN gene, which is the only gene identified to cause Friedreich's Ataxia. Positive genetic test results for Friedreich's Ataxia usually contain 2 numbers that indicate presence of the disease. What do these numbers mean? Our genetic code is spelled out along the double helix of our DNA by triplet combinations of 4 nucleotides labeled A, T, C, and G. At the key place in the FXN gene of a person not affected with the disease, only a dozen or so triplet combinations of the nucleotides GAA would be found. In a person affected with FA, however, this combination of the GAA nucleotides is usually repeated hundreds of times, making it very difficult for the normal part of the gene to be read or transcribed, thus, limiting the amount of frataxin protein that can be encoded and formed. For FA, in 96% of affected individuals, the 2 FXN gene alleles have GAA triplet repeat expansions that silence expression of the gene, preventing enough frataxin protein made for function in the mitochondria. The two numbers in the genetic test reports refer to the number of GAA triplet repeats on each allele copy of the FXN gene. One number is associated with the allele, gene, inherited from mom, and the other number is associated with the allele, gene, inherited from dad. The expanded numbers of the GAA triplet repeats confirm the inheritance of Friedreich's ataxia. Research seems to indicate general correlation between some FA symptoms and the numbers of GAA repeats. In almost 4% of people affected with Friedreich's ataxia, the genetic test report includes one allele of the FXN gene with a GAA triplet repeat expansion and the other allele with a single nucleotide change, called a point mutation, or a deletion in the FXN gene. Changing a nucleotide of A, T, C or G to another nucleotide often causes a change in the frataxin protein structure, thus, causing a change in frataxin's ability to execute its function. A gene deletion removes a section of nucleotides, which can also result in loss of FXN gene expression and frataxin protein function. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about frataxin gene in Friedreich's ataxia. Frataxin, the protein produced by the FXN gene, works in mitochondria to help to create energy. Normally, frataxin works to regulate iron and sulfur molecules in the mitochondria while also working as an antioxidant to reduce oxidative stress. Both of these processes allow the mitochondria to efficiently produce ADP, the source of cellular energy. The nervous and cardiac systems require high levels of ADP, and these two systems are predominantly affected in symptoms of FA. In people with two fully functioning FXN genes, frataxin is made at maximum levels. In those with only one functioning gene, carriers, only 50% of the normal amount of frataxin is made, but they remain asymptomatic. However, in those with two affected FXN genes, very minimal amounts of frataxin protein is made. With limited frataxin, iron-sulfur clusters in mitochondrial proteins cannot form properly, causing a reduction in energy production. Additionally, insufficient frataxin makes mitochondria especially sensitive to free radicals, causing increased damage and destruction of cells. Cells in the brain, spinal cord, and muscles that are damaged or have low energy supplies degenerate over time, causing the signs and symptoms associated with FA. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about clinical trial. A clinical trial evaluates a specific intervention according to a research protocol created by investigators. These interventions may be products such as drugs or devices, procedures, or changes to participants' behavior, for example, diet. Clinical trials compare a new medical approach to a standard approach that is already available or to a placebo. When a new product or approach is being studied, it is not usually known whether it will be helpful, harmful, or have no different effect than available alternatives. The investigators determine the safety and efficacy of the intervention by measuring certain outcomes in the participants. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about clinical trials for Friedreich's Ataxia. While no treatment is currently approved to halt the progression of disease, there are more than a dozen therapies and drugs currently being studied to treat FA. These clinical trials require many steps to ensure the efficacy and safety of the medication before it can be approved for widespread use. Some are still in early investigative phases, while others have advanced to phase 3. For more information on current studies and to learn if a patient is eligible for one of the many ongoing trials, please visit the link in description and talk to your doctors about trials they may be aware of. This information was taken from FARA Australia with their permission. Their link is provided in description.